Welcome to the video lesson on some more advanced genetics topics. In this lesson, we'll be discussing and showing how to do Punnett squares with double crosses, also called dihybrid crosses. And we'll also discuss the concepts of incomplete dominance and codominance. Now that you've had some practice doing some simple Punnett squares, let's give a double cross or dihybrid cross a try. We're going to use an example of this guinea pig here, which for hair color, black is dominant, therefore capital B, and the brown color is recessive and therefore lowercase b. The length of hair on the guinea pig, short is dominant, therefore capital S, and long is recessive, therefore lowercase s. Now in a dihybrid cross or a double cross, what you're going to do is combine hair color with hair length to discover what the possibilities are when various options produce offspring. In our first example here, we're going to cross a male guinea pig that is heterozygous short and brown. Well, heterozygous means you have one of each of the alleles for, in this case, hair length. So heterozygous would be capital S, lowercase s. And so in the genotype, you see that reflected here. For brown, the only way for the guinea pig to be brown is if it had both recessive alleles for that trait. So we have the little b is for brown, and so we see the genotype for brown b, little b, little b. So overall, heterozygous short brown would be big S, little b, little b. We're going to cross that with a female guinea pig that has long heterozygous black hair. Well, the only way to be long is if you're recessive, homozygous recessive, and so therefore the genotype for that would be little s. Heterozygous would mean that you have one of each, so you have a big B and a little b, so we see the big B, little b here for its genotype, and altogether long heterozygous black would be little s, big B, little b. All right, next thing here. When we do a dihybrid cross or a double cross, we use the FOIL technique when labeling the squares. This is the same similar technique that you use in math class when factoring or multiplying binomials. Okay? So let's show you how to do this. All right, we're going to put our male guinea pig here on the top, and we'll put our female guinea pig over here on the left. And now we're going to FOIL the male guinea pig here. FOIL, first, outside, inside, last. So we take the first of each here, which would be big S, little b. Outside would be big S and little b. In our next square. Inside would be little s, little b. And last would be little s, little b. Okay, so our male is all set up. Female first, little s, big B. So little s, big B. Outside, little s, little b. Inside, little s, big B. and last, little s, little b. All right, now our female is set up for our 4x4 square. And that's what, of course, makes these more complicated, is that instead of doing a 2x2 square, now you're doing a 4x4 square. Okay? All right, so now all you do is you take for each square and you combine what you have and express it back to a genotype that includes both alleles for each, in this case, short or long hair and black or brown hair. Okay? Well, here we have a big S, little s. And what I would do is I would recommend to you that you put like letters together, put your S's together, put your B's together to stay organized. And you have a big B, little b. Okay? In this one, we have a big S, little s. And we have a little b, little b. This is tedious, but our work will pay off here. Next, we have a big S, little s. And a big B, little b. Big S, little s. And a little b, little b. All right, a quarter of the way there. Big S, little s. Big B, little b. Big S, little s. Little b, little b. Big S, little s. Big B, little b. And big S, little s. Little b, little b. All right, next row, column, little s, little s. Big B, little b. Little s, little s, little b, little b. And once you get the hang of it, you can really pick up the speed. Little s, big B, little b. And little s, little b, little b. Last column, little s, big B, little b. Next, little s, little b, little b. And you can probably catch on here. This column is going to be exactly the same as our previous column because these two match and all of these match. So let's keep going here. Little s, big B, little b. And little s, little b, little b. All right. Once all that tedious work is done, Now we're ready to go ahead and determine our possible genotypes that we have here and what the percent probability of having those for each one is. Okay? All right, let's just start right here in the top left-hand corner. We have a big S, little s, and then we count how many of those do we have. We have 1, 2, 3, 4. I don't see any others. So that's 4 out of a possible of 16. Well, 4/16 is 1/4, which would be 25%. So we have a 25% chance of having that possible genotype. Okay? Now let's go to a different one. Here we have big S, little b, little b. Then we go ahead and figure out how many of big S, little b, little b's do we have. We have 1, 2, 3, and 4. I don't see any others. So again, that's a 4 out of 16 probability, and that would be 25%. We go ahead and put 25% as the probability for that. All right, next, what other possibility genotypes do we have? We have little s, big B, little b. And how many of these do we have? We have 1, 2, 3, 4. Again, 4 out of 16. So we have 25% chance. Okay? And what's our last possibility here? Little s, little b, little b. And again, we have 4 little s, little b, little b's. And 4 out of 16 is a quarter, which is 25%. Okay? Now for our phenotypes. All right, first short and black. Short is dominant and black is dominant. So the only way to be short and black is if you have a capital S and a capital B. Well, the only possibility out of all our genotypes that shows this is this one right here. So the percent probability of having short black-haired guinea pigs is 25%. Okay? Short and brown. Short is dominant, brown is recessive. So to be short means we have to have a capital S. To be brown means we have to be homozygous recessive. So that would be this category right here. Capital S is short and homozygous recessive for color. So again, we have 25% chance. Long is recessive, so the only way to have that is to have 2 little s's, which both of these do. But to be black, which is dominant, means you have to have a capital B. So we have a 25% chance of having guinea pigs with long black hair. Long and brown are both recessive, so you have to have homozygous recessive alleles for both. So we have to have a little s, little b, and that means we have a 25% chance of having guinea pigs with long and brown hair. All right, now that was quite something, wasn't it? Now we're going to let you give this a try on your own with this example right here. And you're going to have to pause the video here in a moment. And we have a male guinea pig that's heterozygous short and heterozygous black. So that would be a genotype that is big S, little s, and big B, little b. And we have a female guinea pig that has long heterozygous black hair. Long is recessive, so that means she has to have little s, and black heterozygous means big B, little b. So her genotype is as follows. Use the exact same procedure we just did on the previous example, and you should get an answer that looks something like this for your Punnett square. So this would be what your finished Punnett square should look like. And now we transfer that to possible genotypes with percentages. In big S, little s, big B, there are 2 of those of your 16 in the square. Let's talk about percentages after. Big S, big B, there are 4 of those in your square. Big S, little b, there are 2 of those in your square. Little s, big B, there are 2 of those in your square. Little s, big B, there are 4 of those in your square. And little s, little b, there are 2 of those in your square. Well, if 4 out of 16 is 25%, as we discussed in our earlier example, these 2 genotypes would be 25%. Well, what's half of 25%, which is 2 out of 16? Well, that would be 12.5%. Half of 25 is 12.5. So 12.5% for each of the others genotypes that show 2 out of 16 as a possibility. Looking at possible phenotypes, for short and black, remember, to be short means you have to have a capital S, and to be black means you have to have a capital B. Well, we have 6 possibilities for that. To be short and brown, capital S and homozygous recessive, so you have to have little b. There are only 2 that fit that description right there. Long and black is homozygous recessive, so little s, and you have to have a big B for black. So we have 2 there and 4 here for a total of 6 on that phenotype. And long and brown is homozygous recessive for both, and that's little s, little b, and we have 2 for that. Transferring those into percents, we have 6 out of 16, which would be 37.5% for each of these phenotypes, and 2 out of 16 is 12.5%, as discussed earlier, for these 2 phenotypes. You need to ask about this in class if you're not sure and don't understand. And for now, we're going to move on to 2 other concepts. The first one being incomplete dominance. Incomplete dominance is when there is no dominant gene. Heterozygous individuals then show a blended halfway phenotype. The most popular example in genetics for this is the Japanese 4 o'clock flowers, which have red, which will be capital R for us, white, which will be capital W, and if crossed with red and white, we get a blend of a halfway phenotype, which would be pink. Notice that all the letters are capital. There are no lowercase letters when dealing with incomplete dominance. This example wants us to cross a red flower, RR, with a white flower, WW. Well, what would we get? Well, in this one we have an RW. In this one we'd have an RW. And this one we'd have an RW. And in our 4th box we'd have an RW. What do we end up with? 4 pink 4 o'clock flowers, so 100% of our flowers turned out to be pink. Now what if we took those flowers and crossed them with each other? What would we get? So let's cross a pink 4 o'clock flower with another pink 4 o'clock flower. Well, here we would get 2 R's. Here we would get an R and a W. Here we would get an R and a W. And in this box we would get a W and a W. So what do we end up with? Well, you can see we have 1 out of 4 that would be red, 1 out of 4 that would be white, and 2 out of 4 that would be pink. So we have 25% of our population would be red, 50% would be pink, and 25% would be white. This concept is incomplete dominance. We're going to finish our lesson with a pretty neat topic called codominance. And that is, there is no dominant gene. The heterozygous individuals show both alleles expressed fully, but in different places. Great examples of this are the roan horses, which in this, I believe the roan horse on this one is blue and white. You know, it looks black from you. It's actually a dark blue color. Okay, so it has part of the horse that's white and part of the horse that's blue. This roan horse is red and white. So part of the horse has red hair and the other part of the horse has white hair. Calico cats are an example of codominance, where you have the combination of the brown and black fur. That'll do it on our video lesson for a few advanced topics in genetics. More to come as we do more practice problems with all of these topics in class.